Bismillahirrahmanirrahim. 9700 March 24 paper 5 to A-level biology, and the first question will be discussed in this video. This paper is for 1 hour and 15 minutes. It's the same day as paper 2, so you've got to realize that this is more of the marks are much lesser. There are only 30 marks for this, while for paper 2 there are 60 marks. So 30 marks, 1 hour 15 minutes, while in paper 2 it's 60 marks and still the same time, 1 hour 15 minutes. So you can understand the paper 2 is more of an easier paper. It's the AS paper, but here it's an A2 paper. Paper 5-2 is an A2 exam. Bismillah ar-Rahman Rahim. Question 1. Onions are vegetables that are harvested from the plant Allium cepa. In many countries, small onions are used to make pickled onions. Pickled onions can be stored for a long time because the growth of microorganisms is prevented. Pickled onions are made using this method. Number 1. Remove the dry outer layer from small onions. Place the onions in a sodium chloride solution for several hours. Remove the sodium chloride solution and place the onions in a solution of ethanoic acid, which is vinegar. Figure 1.1 shows a container of pickled onions and fresh onions that have not been pickled. So we have the container of pickled onions, then we have a dry outer layer of skin, and then we have the fresh onions. Some students decided to investigate how placing small onions in different concentrations of sodium chloride solution affects the mass of the onions. Identify the independent variable. What is the independent variable? The independent variable is always what you have changed. I always say I independently. What have you changed? What did you change in this? You decided to take different concentrations of sodium chloride. The onion is of course there, but you decided to place the onion in different concentrations of sodium chloride. So that will be the independent variable. So concentration of sodium chloride. Now let's read the rest of the question. The students were given a 20% stock solution of sodium chloride. The student decided to use the stock solution to make a sodium chloride solution with concentrations of 0.0, 1.0, 5.0, 10%, 15%, and 20%. So basically you were given 20% and now you have to dilute it and make 0, 1, 5, 10, 15, and 20%. The students made 140 cm cube of these solutions in separate beakers. So the total volume was 140 cm. Complete table 1.1 to show how 140 cm cube of these solutions could be made by proportional dilution of the 20% stock solution of sodium chloride. You see, when it said complete table 1.1, this was missing here. So volume of what? You have volume of 20% sodium chloride solution in cm cube and volume of something. So what is that? That is what? What could you have said? Distilled water or deionized water? Or you could have said pure water. Just water would not have got you a mark. Now let's add these figures. This is only for 2 marks. This is only for 2 marks. So you had to figure this out is that the percentage concentration and the distilled water. So this wasn't very difficult. If you don't know the formula, then you use the formula M1V1 is equal to M2V2. You need to look this up on YouTube and find out how to do the proportional dilution. We should have done this in the AS level because we do a lot of this in the paper 3 practicals. Now coming to the B part of the question, the students carried out this procedure. The dry outer layer was removed from 30 small onions. 5 of the small onions are placed into each of the 6 beakers. So we have 5, 5, 5, 5, and 6, so that makes it 30, containing the sodium chloride solution prepared by the students. The onions were left in the solution. No volume has been discussed. So the onions were left in the sodium chloride for 2 hours. So we left them at 8 o'clock and by 10 o'clock we finished 2 hours. The mean percentage change in mass of the 5 small onions in each beaker was calculated. That means we must have known the mass initially and then we must have known the mass after 2 hours and then we calculated the mean percentage change in mass. The students had repeated the whole procedure using a new set of 30 small onions. So this is a new set of onions. This time the onions were left in the sodium chloride solution for 48 hours. So we have 2 different practicals. One was for 2 hours and one was for 48 hours. Now, figure 1.2 shows the results of this investigation. Please read this very carefully, understand it, visualize it, and think you're actually doing this in the lab. Now, figure 1.2 shows the results of this investigation. Now, there is a mean percentage change in mass, but you can see the figures are from 0. This is now plus 2, plus 4, plus 6, plus 20. And then there is a decrease. You can see from 0 there's a minus 2 and minus 4, minus 6 and minus 8. So there is a mean positive change and there's a mean percentage positive change and the mean percentage negative change as well. Now it says results of the investigation after 2 hours. You can see the results is the solid line. So it is this line, decrease, then sort of remains less, less, less. Sharp decrease in the from the 0 water to the 5% concentration of sodium chloride. And the other one, which was after 48 hours, you can see the graph how there's been an increase in the water, and then after in the 5% there's a big decrease, and then sort of in a little this and this, and then it becomes constant. So look at this graph very carefully, understand it, and think about it. So there's a negative change and there's a positive change in mass. Now explain why the students calculated the percentage change in mass. Now I always give you this example. If I say 10% of my students got an A, so you can say, okay, well, that means if she had 100 students, 10 got an A. But if I say 2 students of mine got an A, well, you have no idea. Out of how many students? 2 students out of 5? Or was it 2 students out of 20? Or was it 2 students out of 40 students? So I say, okay, 10% of my students got an A. So you say, okay, well, if she had 100 students, then 10% got an A. So there was a time when in my O levels, 100% A's. I had a 10 years record of 100% A's. So explain why the students calculated the percentage change in mass. You see, this allows a very valid comparison. It's a very valid comparison when you have to compare. If I say, okay, I work in 2 schools, school A and school B, and in one school I have 2% A's, and in another school I have 10% A's. But if I say in one school I have 2 students with A's, and another school I have 5 students with A's, okay, by how many students? Out of how many? What was the total number of the class? So this doesn't tell you much. So allows a valid comparison. Allows comparison as the initial or the starting mass of the onions is not constant. It's not all the same. 5 onions may weigh 2 grams or 3 grams or 2.5 grams. So any of these answers was correct. Any one of this answer was correct. Part 2, one of the students concluded that the water potential of the onion cells is the same as the water potential of a 4.2% sodium chloride solution. With reference to the information provided, including the results shown in figure 1.2, suggest reason why this conclusion should not be accepted. Now, let's look at the graph again. How do we find out the actual percentage of this thing? You see, you have to find out where it cuts the 0 mark. This is the 0 mark. Now, one of these, which was kept for 2 hours, is somewhere here. So it's nearly 1 and 2, 3, 4, 4.2. Yes, this is here. This is 4.2. But this is only for the one which was kept for 2 hours. What about this other one? Now you can see this is somewhere near 3.1. This one here. So the one which was kept for 48 hours it intercepts the zero mark at 3.1 percentage concentration of sodium chloride. So how can you say it is 4.2? Now let's look at the question again. With reference to the information provided, including the results shown in figure 1.2, suggest reasons why this conclusion should not be accepted. The conclusion is only supported for onions left for 2 hours. And where are you going to check it? Is the 0% change in mass. And the one left of 48 hours, that is 3.1% sodium chloride. Then line of best fit may give a different value. So there are no intermediate concentrations between 1% and 5%. We should have taken many different concentration between 1 and 5%. Then no statistical analysis has been taken out. And then not all onion cells have the same water potential. So conclusion supported only by onions left for 2 hours, and then 0% change in mass is 3.1% sodium chloride in onions left for 48 hours. Then intermediate concentrations have not been tested. We only have 0, and then we have 5%. So we don't have anything between 0 and 5%. You see, this is the 0 and this is the 5%. And we don't have any values in between. Then no statistical test has been carried out. Not all onion cells have the same water potential. And then there could be some anomalous results on the graph. There could be an experimental error in this. There could be an idea that the line of best fit may give different values. So some of the points I've not written, but I'm just telling you about this. So you need to think about all those points. Part C of the question, the turnip Brassica rapa is a root vegetable. Turnips are grown in many parts of the world. Figure 1.3 shows freshly harvested turnips with the leaves still attached. Now in Urdu that will be called shaljam, turnips. Turnips, root vegetable, leaves. Now it says the student decided to investigate the effect of temperature on the rate of osmosis in turnips. In an initial test, the student removed the outer layer from a turnip and cut the turnip into small blocks. The student placed one of the turnip blocks into a beaker of distilled water. You see 100 centimeter beaker, distilled water, the volume is not mentioned, and the turnip block. As shown in figure 1.4, water entered the block by osmosis. Fine. So this is the block and water will enter by osmosis from all sides. The student was provided with additional turnips and standard laboratory apparatus. Describe a method that the student could use to investigate the effect of increasing the temperature over a range of 10 degrees to 50 degrees Celsius on the rate of osmosis in turnip blocks. Do not include the risk assessment or how to calculate the rate of osmosis from the result. Method should be out in logical order and be detailed enough to allow another person to follow it. Now, usually I tell you the risk assessment, I always tell you it's a mark scheme point. Now they said do not talk about it now. So usually I always tell you when there's a long question, give me the hazard, risk, and precaution in this manner. Give me the hazard, then the risk, and then the precaution. What would it do? Like for instance, like I always tell you about this is that I'm allergic to mangoes. So if there was an experiment on mangoes and I was touching the mangoes, well, I would get a rash immediately. So if somebody is allergic to turnips, then the risk is allergy. The hazard is turnips. Allergy, use gloves. So hazard, risk, and precaution. Now they have given you a clue in the earlier part of the question is the weighing the mass of the onion. So they gave you that clue in the first part of the question, and so you will just take it from there. Now what will be the main thing? 5 stated temperatures. So you take 5 beakers and then you decide, okay, how are you going to have it? So 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, 40 degrees Celsius, and 50 degrees Celsius. Now you'll maintain them in different water baths, the beakers. So use a water bath, a thermostatically controlled water bath to maintain the temperatures. Then the turnip should be the same age or the same variety of turnips. Then the same size of the turnip block, that should be the same every time. It shouldn't be different. So same size of turnip block in each. Maybe I don't draw it very exactly the same, but I mean to be exactly the same. So same dimension of the turnip, same size or same dimensions of the turnip block. Then how would you get it the same size? This is another thing. How, what would you use? You would have to use some sort of a template, like you have these cookie cutters, or a bottle top is sometimes used as well. So you would use some sort of a cutter to really cut the same size. Then for each temperature, place at least 3 blocks in each beaker. That's what we always say that. So for each temperature, place the block in a beaker and add fresh distilled water, and that should be the same volume in each. Right? So then how are you going to measure the rate of osmosis? So measure the initial mass and then the final mass after say 2 hours or 3 hours or 4 hours, because we are measuring the rate of osmosis. So at each temperature, measure the initial mass and then the final mass after a set time, or you can say 4 minutes or 5 minutes. And for each temperature, use at least 3 different blocks and calculate a mean, because that increases the reliability of the experiment. But as you take the block out, as you've taken the block out, then you need to also think of removing the excess water from the block before measuring the final mass. So you need to use some sort of a tissue to see that the extra water around it has been removed, because that would affect your final mass. So you sort of use a tissue to pat very gently, not forcefully, pat very gently and remove the excess water. Minimum 5 stated temperatures. Use thermostatically controlled water bath. Same variety of turnip. Same dimension of the turnip block. Use a cutter to cut same size of the turnip block. You can use anything if you want to say apple corer or something like that. At each temperature, add turnip block to a fixed volume of distilled water. Measure the initial mass and the final mass after a set time, maybe 2 minutes, maybe 3 minutes, but in the previous experiment was 2 minutes and 48. So you have to think of that, something in between that. Measure initial mass and final mass after a set time. Then use at least 3 blocks at each temperature and calculate the mean and remove excess water, pat dry using a blotting paper or something like that. Now coming to part 2 of the question, complete the sketch graph shown in figure 1.5 to predict the effect of increasing the temperature over a range from 10 to 50 degrees Celsius on the rate of osmosis in turnips. Include axis labels with units in your answer. So here it would be temperature in degrees Celsius, and here it will be rate of osmosis. Because please do not add mass, because that would be incorrect, because we are investigating the rate of osmosis in turnips. So it's a gradual increase. So how would you get the 2 marks for? Temperature in degrees Celsius and vertical axis rate of osmosis, and the graph is, of course, the temperature increases, the rate of osmosis will also increase. Then coming to the last part of the question, suggest one hazard of your method in C1, the risk associated with the hazard and the precaution that would need to be taken. Now hazard could be anything. The knife could be a hazard, so risk is injury and cut away from the hand, cut down onto a board. The other hazard could be the turnip. What would be the risk? You could be allergic to it, or it could result in an irritation and wear gloves. The other hazard could have been hot water, water bath, and this could burn you or scald you or injure you. and wear heat resistant gloves and use tongs to pick up the turnip blocks. So I've only given one of these, turnip and the allergy and wear gloves, and this is all for 1 mark. But you could have said any of the other ones. You could have said the knife cause an injury, so cut away from the hand, and hot water, so water bath could burn you, and you should wear heat resistant gloves. Thank you very much for watching, and the question 2 will be followed in the next video.